Hello everyone, I hope you all are good and doing study well. Myself Priyanka Gupta from Dr. Hari Singh Gaur Vishwavidyalaya, Research Scholar in Department of Zoology. In this video, I am presenting an overall idea about the biostatistic tools useful in the biology. As you all know, there are many statistical tools and software tools such as SAS, SPSS, Stata and Minitab etc. which are widely useful in the biology. But today I am going to describe only statistical tools useful in the biology. So, this simple flowchart talks about which type of statistical tools useful in the biology. It all depends on what type of data they are. Is it continuous or discrete or the categorical data? Now question arises, what is the continuous, discrete or categorical data? So, the data is said to be continuous if the values are within a finite or infinite interval, or in simple words we can say measurable values. For example, wing speed, volts of electricity, etc. And a set of data is said to be discrete if the values are belonging to the set are distinct and separate, or simply we can say countable values. For example, number of votes in an election and number of eggs in a basket, etc. And when numbers are collected in groups or the categories is said to be categorical data. Now, if the data is discrete or categorical, we use chi-square test. Why? In general, this test we use to measure the differences between what is observed and what is expected according to an assumed hypothesis is called chi-square test. This is a non-parametric test, means it is based on frequencies, not on the parameters like mean and standard deviation, and used to test hypothesis, not useful for estimation. Next, if data is continuous, then we have two statistical questions. First is there difference between mean, and second is there relationship between two variables. Now we come on first statistical question. If there is difference between mean, then how many treatments we have? If we have only two treatments, so we will apply t-test, or if we have more than two treatments, we will apply ANOVA. First is t-test. It is a parametric test, means based on parameters such as mean, variance. It can be either paired or unpaired t-test. Now, what are the assumptions for applying t-test? Random samples are drawn from normal population and the samples must be less than 30. Now, if we have more than two treatments, we will apply ANOVA. Why? For determining if differences in means are significant and for dividing the variance into components. Certain assumptions are followed in the analysis of variance. First, the samples must be drawn independently. Second, population are normally distributed. And third one is effects of various components are effective. Now, we come on the second statistical question. Is there relationship between two variables? So, why we use linear regression and what is it? Here, the dependent variable is a function of a single independent variable. For example, height of a person on his weight is linear relationship between two variables. Now, can you see this beautiful picture? This is the proper way to conclude this presentation. First, chi-square test deals with categorical data only and based on comparison. T-test deals with categorical and continuous data both based on comparison. And the linear regression deals with only continuous data based on relationship. Thank you all for watching and listening this presentation. Thank you. Stay safe, stay strong.